So here are a group of individuals. Look at the babies or the kids. None of them look similar to another. And even in the case of adults. In fact, when you go into a train and if you see it, every person next to you is different from each other. When you walk on a street, every person next to you is different. Well, of course, unless you are a twin, that's a different scenario altogether. So what is this reason? Why is there so much difference? So much what? In fact, there is complete difference from one individual to another within the same species of human beings on the same planet Earth. What makes such uniqueness? What is the reason behind that uniqueness? Well, the answer to that question is within ourselves. What is within ourselves? Let us put it down. Within our body, there is tissues, right? And these tissues are made up of cells. And the answer lies within each and every cell in a specific region of the cell called nucleus. Okay, so what is present in this nucleus? That one, that fellow who is waving at us. Now that person or that fellow is nothing but our genetic material. It's nothing but what we simply call as our DNA. The genetic material of us is nothing but our DNA. Let's write it in DNA, the deoxyribonucleic acid, which is part of what we call as our genome. Right. So this fellow that is waving us, it's the genetic material. This fellow is the one that is responsible for the changes or the uniqueness that is present between one individual to another. Whatever differences we have from one human being to another, it is due to this DNA. Now, if I have to compare DNA, I can compare it with a cookbook. Now, why should I compare it with a cookbook? See, let us say a cookbook has, what do you have it? You will have ingredients, you will have recipes, you will have quantities of what to put, how much to put, so that you get your final tastier recipe. Correct? Similarly, DNA has information. Information about what? About proteins. Okay, now what should be present in this proteins? Now let us say one such protein, we'll call it as our hemoglobin. Now hemoglobin, it should contain 2 alpha globin chains and 2 beta globin chains. Now this information that a hemoglobin protein should have alpha chain and beta chain, that should be, their number should be 2 and 2. This information, everything is present on the protein or on the DNA. Along with that, you will have information for other structures like RNA, like tRNA, when they should form. And when we are talking about proteins, hemoglobin is a structural protein. There are many such proteins, right? So all that information is present on the DNA. In fact, if we have to compare it, we can say that DNA is like the blueprint. I mean, what does a blueprint have? We have all seen blueprints, right? It is present with our architects. Now, in a blueprint, they will have the small miniature version of the house that they are going to build for us. Like where should be the hall, where should be the dining hall, where should be the kitchen, where should be the bedrooms, and where should be the playroom, everything. All that information of where what should be present in a house even before it is constructed is in the blueprint. Similarly, everything about a human being, every factor about the human being, like these are some rough factors that I'm mentioning: the skin color, then the hair color, the eye color, then the hair texture, physical attributes like height, physical attributes like basic weight. Okay, so all these information, all these particular information of how a newborn human being should be, or how this particular person will be throughout the span of his life, that is present already in the DNA, and that's why we call DNA as the blueprint of life. Now, in this DNA, when you take any human being's DNA and when you subject it to density gradient centrifugation, density gradient centrifugation. Okay, so where you are centrifugating the DNA molecule against cesium chloride, and when you do it, the DNA molecule gets separated based on the density. It is as simple as that. So when you do it, you get, and when you plot peaks about it, you get two peaks like this. One peak is called as the bulk peak, and the other one is called as the satellite peak. Satellite comes from the Latin word satellus, which is nothing but the subordinate or the other one. So if you consider this as the main peak, this one is the subordinate or the second peak. And we found out that the DNA sequences in the bulk region were completely common sequences, whereas the sequences within the satellite region, they were unique. So with this, we understood that our DNA or any human being's DNA or any eukaryotic DNA is divided into two. The 99% region, which is the majority, and the 1% which is in the minority. The 99% is called as the bulk DNA, which we share with one another. And this bulk DNA is common. There is no difference in another. As I said, when you sit next to a random stranger, even though he's a stranger to you, he's completely different looking to you, you should know that internally you both share the same 99.9% of your DNA between both of the same is both of you is same. The only difference is this 1% or 0.1 to be very specific, which creates that uniqueness. That main differences among us is created by this 0.1%, which is the main ideological one. So it is a very small amount. It is not a huge amount, but that small amount is capable of making such a huge difference where every individual is different from another. Now, in our globe, as we have said, there should be many unique sequences in that matter. Let us say if our Earth is made up of 7.6 billion people, hypothetically, then there should be 7.6 billion unique sequences, because in a population, every person shows unique sequences. Okay, and these unique sequences are called variable number of tandem repeats. Now, where is this VNTRs present? Now, let us understand something. The phenotype, you must have studied this in the genetic chapter. The phenotype, sorry, the phenotype is something that comes because of the genotype, which is nothing but the sequence of alleles in a gene, and it is the phenotype that attributes the physical characters. Now, when we say gene, you must have seen it in the structure of the gene. You must have understood that a gene contains coding sequences which actually code for the proteins which form the phenotype, and between the coding sequences there will be many non-coding sequences. Okay, so we'll put these C's as the coding sequences, whose ultimate result or end products will give you the phenotype, and the rest are the non-coding sequences. Okay, previously it was thought that these non-coding sequences are completely useless. They used to call it as junk. But now you know that these non-coding sequences actually contain the VNTRs, and they are also responsible for the uniqueness as much as the coding sequences. So let us understand VNTRs once. Now, if I have to break the word VNTR, if I have to break it, I'll break it in an interesting way. Okay, so let us break it. Variable, number, tandem, repeats. So when we are talking about the non-coding regions, let us put it here, the non-coding regions, there will be repeat sequences. Repeats means what? Let us say there are three base pairs, A, G, C. If these are continuously getting repeated like this, AGC and AGC, so this sequence is called as a repeat sequence. Okay, And when we say the word tandem, tandem means what? They are adjacent, means they are side by side. So you can clearly see here the repeats are present side by side. There can be another way where two repeats will be here, then there will be another sequence, another repeats here. So this kind of scenario can also exist. But if they are present side by side like this, we call them as tandem repeats. Then what is this variable number? Well, variable number means the number of repeats varies from one individual to another. How is it possible? Let us see. So VNTRs are a short sequence of bases in a DNA that is repeated at specific chromosomal locus. So on our chromosome, you have the gene, and we have mentioned already that the gene contains the coding and the non-coding region. So in the same loci, you will have the repeats whose number will vary from individual to individual. So let us say this is one human chromosome and this is the location of the VNTRs. Now, if I take the same chromosomal locus in three different individuals, let us say individual 1, individual 2 and individual 3. In one, the number of repeats are these many. In another, it is this many. And in the individual 3, there are only 3. So, the locus is the same. The locus is same. The chromosomal number is the same, but what is very different? What is the variation? The variation is the number of repeats. So the number of repeats, VNTRs are regions which vary from one individual to another. So you can call VNTRs as similar to alleles because they are variants. So we call it as allele variants, and they are similar and they are completely different. Their number is completely different from one individual to another, and that is why they are called as highly polymorphic. Polymorphic means they have lot of changes. The capacity to have multiple variations is called as polymorphism. Poly means many, morphs means different varieties. So because you are having, each individual will have a different specific number, these VNTRs are said to be highly polymorphic.